Welcome you all to today's event on the compelling theme of One Health for the Well-being of All, under the aegis of G20 Chief Science Advisors Roundtable.
If you see, One Health is a collaborative, multi-sectoral, and multidisciplinary approach which actually covers human beings, animal, environment, plant. The entire ecosystem is what is encompassed by the whole philosophy of One Health. And therefore, the approach to human health in isolation and not looking at in the context of what else there in around is not really valid anymore because changes in environment is impacting human health and the zoonotic jumping, which has happened, in fact, COVID has been a big example, is also a reason why we should be bothered about our relationship with the animal kingdom as well. Because this jumping of viral infections or even bacterial infections opens up some kind of new directions to explore and look at. Look at in terms of creating also a strategy for prevention. So the environment, if you look at today, linked with One Health, because the data is fundamental of digital health. The only challenge today is that in Indian system as well as globally, the data is fractured, it's not organized, it's not available. And that is a major bottleneck for really building up a One Health strategy. Because without data, you cannot create these models. We live in a world where human beings, environment, animals, plant, everything is linked together into an overall systemic fashion. Ability to understand and model the system and based on that, creating health strategy is need of the day. And another very important thing that we are looking at today is in terms of integrative medicine, integrative precision medicine. Because there are different schools of medicines and thoughts. We need to analyze scientifically, we need to discover their fundamental bases and try to see that how well we can use those concepts because in many cases, those traditional medical sciences also look at human being as an element of an overall environmental system and not just in isolation.
Work because as it is all about the ecosystem health and biodiversity conservation where there are so many interdependencies and it has to have a systems approach in trying to understand the problem and there are challenges of scale. We cannot do it at a small piecemeal level. It's just not possible. There has to be a context, socio cultural, spatio temporal, because those kinds of things can totally change the ecoregion in various ways and you cannot actually port information from one to the other. And there are multiple levels in which you need to assess the health of the ecosystem, both below the soil, above the soil, and within us, within the fauna, within the flora. So it's because everything is seamlessly connected and anything perturbed in anywhere will be reflected as an indicator in any one of these places. But the question is, how do you collect all this, right? And it needs to be economically viable and sustainable. It cannot be one-time exercise. So it has to be in such a manner that you are able to address this. And then when we come to the implementation, that's why we need transdisciplinary involvement collaborations across multiple stakeholders, each have a very important role to play in not only getting the best of the science, best of the innovation, and also sustainability. So we need to connect with academia, research organization, health, environment, agriculture, veterinary, strategic sector, and then also because those are the people who are going to be the final beneficiaries, and if we don't connect with them, we are not really aligned to the needs and to also the way the research should be oriented, industry and NGO. And all these should actually help in development of a knowledge hub, which is eco-region specific, the context specific. For that, we need to build capacity, because we just cannot create knowledge without capacity, and we need skills. So, and we need technology to be able to upscale it, to be able to disseminate it. The other thing which is very important is benefit sharing. This has to have a equity in such a manner that it is inclusive. It is not benefit to one particular set of stakeholders. It has to connect to the community, and a development of recommendation engines which could allow, just like Professor Chaudhary mentioned, early actionable interventions, remediation, and rewilding solutions, and also create evidence which would enable policy recommendations. So this is the framework which we need to develop in any context, and these all considerations have to happen. And as Sir mentioned, the clusters which have been set up by the Prime Minister's Scientific Advisory, these provide a very, very amazing enabler for setting up these things. Like the Jodhpur City Knowledge Innovation Cluster, we are connected and we are getting more and more connected through this cluster. It enables to source expertise, to reduce on the time to develop technologies, to reduce the cost, because lots of things we will not be should not be able to incur to develop things. And also it provides connects. Like Thar Design is one such example. Thar Design is Desert Ecosystem Innovations Guided by Nature and Selection, which was a pilot study in the Jodhpur cluster. And through that, we could actually organically develop it into a One Health perspective. And very slowly, we could actually connect a huge number of people. And this is like still a networking is getting built. And today, through some of these networks and some of the networks of these networks, we will try to see how we can, at the end of the day, maybe come out with certain kinds of knowledge, certain kinds of key points which would enable us to have a focused way of growing in a particular direction. So the idea is today's is that we kind of, if we can think through a strategy for meeting sustainable development goals, because there has been an original state which is not there anymore. Development, anthropogenic activity, climate change, all of them are contributing to the present current state, which we now know is not going to be sustainable. So we have to actually move towards an envisioned state which has to be in a One Health perspective. And all these points which are needed to be done, it requires multiple stakeholders to come to a platform, and the government to be able to support such kind of initiatives.
Animal vaccines, I visited that it is much better than human vaccine ones. They create all the important animal vaccines. So there are issues, there are means and others to tackle One Health through the data, through the new spatial geography things, hydrology and other things, environmental monitoring, chlorophyll monitoring, the global warming, the temperatures around to which the dengue is really related. So there are issues, but the human behavior is one of the major issues. It is very difficult to change human behavior, even our own behavior. So this is something which should be frontally tackled.
Actually, you gave the example, take up the example of pesticides. Pesticides continue to be used, but then what alternative? If you are synthesizing pesticides, why don't we invest heavily on the research which will lead to degradation, after a shorter while? Actually, if we synthesize so many chemicals, about whose disposal we are worried about, why we don't work on the chemistry which leads to the same distinction of the same chemical? Actually, So the point arises, the science for future also has to be driven both ways actually. Synthesis and distinction. Synthesis and its own degradation. And there we stop.
One Health is a collaborative, multi-sectoral and transdisciplinary approach, as well as interdisciplinary approach. So keeping that in mind, let's talk about how where these interactions come in and what is the importance of these interactions. Wild animals have an infection cycle. There are low levels of infection spreading throughout those populations. And if you have an infection, you also tend to shed those infectious particles. Now, in some cases, those infectious particles may shed to a bridging host. They could be a domestic animal, or they could shed directly to humans. And that can result in a spillover event. Okay. Now, spillover events happen all the time, but sometimes the infection itself, as a result of that spillover, happens rarely because there's lots of barriers to cross before a pathogen can actually, from animals, can actually infect humans. The spread cycle is what's interesting and what's got us most interested recently, because the spread cycle is often independent of what happens on the left side. The spread cycle is often the result of modern human activity. So travel, urbanization, global travel, so on and so forth. And there's some broad patterns to the risk of emerging and re-emerging infectious diseases. Okay. Several scientists have come together to analyze what these patterns tell us. One is that areas that are biodiversity hotspots, so basically where you have more animals and more life, is also where you're likely to have more organisms that can be pathogenic. And of course, as humans, where people live is where we are most susceptible to those pathogens. And where livestock occurs, because where people live, we also have a livestock, they can then form that interface more closely. So those then result in hotspots of risk of emerging and re-emerging infectious diseases. In a sense, what we want to do then, using a One Health surveillance system, is to try and understand what those different boxes are that we want to target. Now, One Health is traditionally, at least in the Indian context, we haven't really embraced it in its entirety, mainly because we are focusing on either the animal side or the human health side. We're actually looking at the after effects of it. I'll give you a couple of examples. Now, this is a study that we've been doing in Pune city. Rabies is a really common disease in India, but rabies control has almost exclusively focused on the human side. So post-exposure prophylaxis. Very little focus has been given on the animal side, and what little has been given in terms of mass dog vaccination and so on is again devoid of science from the Indian perspective. And they run an animal rescue facility. So we adapted their system into a passive sampling survey, passive surveillance system for rabies. We found that the number of rabies positive cases in Pune is quite alarming. And lots and lots of cases. Okay, almost more than 700 plus cases. And more or less equal throughout the year with over the years with some sort of seasonal pattern of emergence, with some hotspots in space, but not necessarily in time. What it tells us that where people are living with animals is always a high risk of disease. Okay, and it's important for us to also study the animals in great detail, study the ecology of the animals in great detail to be able to come up with ways of preventing that spillover or of ensuring that spillover doesn't occur, or if it does occur, we are at the forefront. So in the case with Pune city, whenever we found a rabid positive animal, we sent teams there to educate the people around that area. And thankfully, no cases of rabies were reported from Pune city itself in all of those 4 or 5 years. Nobody's looking at the rural areas, whereas 95% of human rabies cases in India are reported from rural areas. Something like new figures need to come up, but previous figures indicated about 20,000 human deaths. People depend on forest landscapes for their livelihood. Millions of people do. People depend on animals for their livestock, for their livelihood. Okay, we have the highest number of livestock in the world. So to ask people to reduce their interactions with wildlife, or to reduce the interaction between wildlife and domestic livestock, is almost impossible. So what can we do in this regard? How can we optimize benefits that forest dwellers have whilst trying to reduce the impact of diseases? Sort of went back to the drawing board. Now, this disease is a tick-borne disease, okay, and it's caused by a flavivirus that can cause fairly debilitating hemorrhagic disease. At some point of time, more than 500 cases were reported, but those number of cases have been coming down of late, primarily due to a lot of efforts by the government in trying to educate people about how to reduce their risk from this disease. But the disease has also been spreading in geography. Okay, and it's a very complex transmission cycle, as almost all vector-borne diseases are. You'll see that the ticks themselves have a multi-step life stages. It is finding a host for a blood meal, because those blood meals are important to produce their eggs or to move on to the next stage. Okay, and the amplification of the virus can happen at each of these stages. So we found KFDV in all of those different hosts. They're found in small mammals, they're found in rodents, and they're found in humans, of course. Humans are an end-stage host. They don't transmit that virus further. Livestock don't tend to get the virus, or we haven't found enough evidence that they have high viremia if they get it. So they also don't play a role in transmitting of this virus further, but they do play an important role in tick number amplification itself. Sampling all of these and trying to understand these different stages really requires this interdisciplinary One Health surveillance, where you're not only sampling for ticks and all the domestic animals and catching wild animals to study which species have what and where, not just what, but where. What kinds of habitats are riskier for these kinds of diseases or for risk of catching ticks. But you also have to understand where animals move. So you deploy them with, you put GPS collars on them, follow their movement around. But eventually we are concerned about what happens to people. So we also do people surveillance. We try to understand their behavior and what happens when they move into the forest, what happens when they are doing their normal routine, and which habitats or which landscapes are riskiest for them in terms of catching ticks. Okay, and that way we can develop better systems of trying to prevent people from getting infected, even in the absence. So Dr. Ganguly spoke of vaccine. That vaccine, unfortunately, has been found to be ineffective now. It was an old technology. There was no improvement of the vaccine. The vaccine has been discontinued. They're trying to develop a new vaccine now with more modern technology. But at the moment, people are unprotected. So we need to use other methods now of reducing their susceptibility to the disease. So we have come up with different tools. So for example, a decision support tool for trying to predict where zoonotic disease can occur. Now, the problem is that most of these tools focus only on single set of processes that drive this risk. They often, it might be only a social process or an epidemiological process, or very rarely an ecological process. But the interdisciplinary nature of this work means that you are able to then link all of those together in a proper One Health framework, okay, that then allows you to understand what is disease risk. And that's what we came up with for this landscape, where we used all of the data that we had collected, and we were able to then come up with a spatial model, a spatially explicit model of where KFDV can occur in the landscape. Okay, and then we build these models that will allow us then to predict ahead of where the next outbreak can occur. So I'm just going to end my talk very quickly with this last slide, where we opened up to see if this is actually happening at at the level of India or not. And what we found is that there's a lot of institutions in India that work on zoonotic diseases. Okay, most of them are from the biomedical field or from the animal health field. And you see there's one corner out there, the wildlife health field, which is barely connected to the rest of them. Okay. And that's the gap here. And this is what we really need to address. Ecology is currently missing in the field of One Health in India. And ecology, in fact, has to be at the center of the One Health field, because after all, that's what all the interconnectedness really is in One Health. So
environment, then we know that water, air, soil, and biota constitutes the four most important components of the environment. My probability of catching a disease by in this particular room is directly proportional to the distance at which I'm sitting with a person who has an infection. But my probability of catching a disease because of polluted air is much higher because we all are inhaling the same air in this room. We all are consuming the same water that is being supplied to us. So water, air, soil is something that impacts us at a much greater scale. And of course, that doesn't mean that the health angle is not important. Absolutely, extremely important. But if we look at the root cause of the problems, then it will not be an exaggeration to say that environmental health maybe is at the apex of causing the entire problems or the crisis that the human populations are facing these days. So these two examples which I took, one is climate change. We know how climate change is impacting biodiversity, and this very loss in the biological species has led to zoonotic diseases, scarcity of water, and human encroachment. Environmental contamination, for example, soil contamination, has the ability to impact soil fertility, which can result in the production of inadequate food, can cause malnutrition and emerging diseases. So a lot of works are happening around the world on development of advanced treatment processes. And this is the time that we adopt those processes if we are seriously thinking about One Health. This particular slide shows the water contamination in the conventional wastewater treatment system. So as we can see that when the water goes down our drains from our toilet flushes and our bathroom sinks, it eventually undergoes three types of treatments in a conventional water treatment system. So the primary treatment, which essentially is physical, that separates the solids from the liquid, and the secondary treatment, which is essentially biological, where the microorganisms convert the chemicals which are present in the wastewater into the harmless gases. The primary, secondary, and tertiary treatments are the ones which are responsible for providing clean water in our taps. This entire process is highly energy intensive. So as you can see, there are raw sewage or the raw wastewater. It's taken to the screens, to the grid chamber where the solids and liquids are separated by sedimentation. The treated water, the solids are separated, and the liquid part, which contains the soluble impurities, then enters the secondary treatment where tiny microorganisms degrade the water. A lot of energy is spent in aerating the water, in providing the oxygen to the water to get rid of the wastewater constituents. The wastewater is treated and the insoluble bacteria are produced. These bacteria are also the major ground of holding because it's a consortia of microorganisms. There are a lot of pathogens, and therefore a tertiary treatment is done to disinfect the water before providing us to our taps. So while this process is happening, as I said, it's extremely energy intensive, requires very high energy input. It is in the order of 0.3 to 2.1 kilowatt hour per cubic meter. We have to really invest a lot of energy. There's improper utilization of solids and sludge. The solids that are separated are eventually ending in the landfill. We are not making use of that. The long treatment times, one single batch of water takes 10 to 15 days for treatment, and the systems are fixed volume. So this is a picture which shows the typical wastewater treatment plant, and you can see that these are all concrete-based structures which have fixed volumes. They cannot really undertake increased demands or increased flow of wastewaters. If the flow of wastewater increases, the capacity of the treatment of the plant automatically goes down because the systems are not modular, the volumes cannot be increased. So the inability of these systems is also to remove the recalcitrant or difficult to degrade compounds. And the other important thing with the conventional water treatment systems is that the water quality monitoring relies heavily on the lab tests. The water is taken to a laboratory where all the water quality parameters are monitored. Similarly, there is a water distribution network failures that also causes a problem with with getting the water through our taps. So adopting modern solutions can really help us in overcoming some of these difficulties. For example, wastewater has 10 times more energy than what we are investing in treating it. If we use the energy which is hidden in the wastewater and harvest that energy, it rather turns the entire cycle into our favor. We don't have to invest energy in treating water, rather we harvest energy by treating water, and while we do that, we also treat the water. Having centralized versus decentralized water treatment systems. If you have a common treatment facility for the entire city, it is very difficult for the water to transport to that particular treatment plant and reach back. But if you have decentralized systems, then that can really help a community, a population, a city, a society to have water quickly through its treatment plant. We can make use of high-rate bioreactors. A number of researchers across the world, including our institute, have developed processes which are very high rate. They can treat the water at the rates they can meet the demand of the population. Adopting those systems also helps with lesser sludge and energy harvest. Constructed wetlands is one example. Advanced oxidation processes is another example. Then the most important thing is the intelligent sensor systems for smart water management. Water that is currently supplied is of a single quality or a single grade, whether it is coming through my bathroom tap or it is coming through my kitchen tap. It is important to grade water, and that can also help reduce the amount of energy that is invested in treating the water. So for smart water management, a number of sensor systems that can be installed to grade the water where the role of IoT and such networks come into picture. Water surveillance for the transmission of waterborne diseases again become a very, very important component of water sensing system and the creation of early warning systems, as we have been hearing. So just to give one example that we've been working on, of course, there are numerous examples, as I said, but then we can harvest the energy from the wastewater, and we can actually compare that with the conventional processes to see how it weighs over the conventional systems. So if we look at microbial fuel cells, for example, the microbial fuel cells are capable of harvesting energy from the wastewater. They are capable of treating different types of wastewater. If I mix industrial wastewater with the municipal wastewater, there is a probability that the entire system fails because industrial wastewater has toxics. But when we have a robust system that is adaptable, then we can actually mix a composite waste and treat that in a microbial fuel cell. These systems can harness the energy from the waste rather than investing the energy towards the water treatment. Then if we compare the conventional process with this modern process like microbial fuel cell, we can see that the conventional process, it removes carbon, nitrogen, and phosphorus sequentially. And that is why it takes 10 to 15 days time to treat a batch of water. Whereas a system like microbial fuel cell can simultaneously remove the three nutrients. The conventional process needs energy input. The MFC process can produce electrical energy, and it can also be combined to produce biohydrogen or biofuel. So this entire water, energy, and food nexus can be taken care of if these advanced processes are adopted. So these processes can also help with not just the water and energy nexus, but also the water, energy, and food environment nexus that optimizes the usage of energy across the treatment processes. Another example that I have is of the sensors and AIoT-enabled water monitoring and distribution system. The conventional distributions have problems because the water supply rates are lower because the treatment times are longer. The higher treatment costs are there, but with the use of surveillance system, with the use of sensors and networks and grading the water, for example, the nitrogen and phosphorus-rich water can be supplied for agriculture. In that case, one can save the cost on the fertilizers. Lower energy consumption because we now don't have to treat the water completely. Also, it meets the demand and supply by lowering down the energy input into the process. So with that, Environment surveillance, as I said, environment lies at the apex, and it's very important to take care of the environmental health, because once I take care of the environmental health, I can take care of the number of crises that are arising because of the One Health. And the consumption and the production activities have to be balanced. Currently, our consumption activities are producing a huge amount of environmental impact and stresses on the ecosystem. In order to conserve that, we need to really get into resource discovery and minimum disposal, like in circular economy, and these advanced oxidation processes and advanced water treatment processes, where a lot of life cycle analysis have been done on case-to-case basis. If these are adopted, then they can help restore biodiversity, they can help control pollution, can help stop pandemics. We will listen to numerous examples in the morning how different type of zoonotic diseases are emerging. But then if we take care of the root cause, probably the ones which are in line, probably we don't know about the infections which are probably ready to infect us, or the pandemics which may come down the line 5 years, 10 years, may be stopped. We can reduce economic stress, enhance food productivity, ensure human and animal health, and also conserve natural ecosystem. With this quote,
carbon sinks. So a lot of things. So if I were to just simplify things, whatever HP's printer prints over the next 10 years have already been counterbalanced today as a I would say as an initiative, which over 10 years would one counterbalance, and after 10 years, we believe these to act as carbon sinks for a lot of other GHG emission reduction projects which we offer to our clientele. These are projects which we call carbon neutral computing, where third-party assured carbon neutral certificates are provided to our customers that their entire carbon footprint of their product, right at its production, transportation, and product use over the next 4 years have been, I would say, offset. And that is something which we provide as carbon neutral computing services. Okay, these are some stats. And this is something which we also believe that what we're doing within our own premises need to be communicated to be able to create a change-making effort or influence others to adopt what we're doing, and actually conveying that it's all a lot of business sense.
So paramount that it influences everything that has to happen under the One Health. And you've seen since morning, a lot of these speakers spoke about all the diseases that come from maybe wildlife because of displacement, or because of zoonotic diseases, because of livestock. That's part and parcel of agriculture. So, but farming community, I can tell you with my very vast experience that I had for many years now, is changing quite rapidly with quick adoption of technology, innovations, regenerative practices in agriculture, to the extreme of genome editing, to the recombinant DNA technology. Farming community are adopting drone-based technologies. We got into the precision agriculture with all kind of micro, macro irrigation systems that are putting in place in agriculture. Farmers are also moving to decarbonizations. Farmers are also moving to areas where they can sequester carbon. We are also looking at post-national trade in agriculture that is close to $2 trillion. And our contribution, which was never been more than 15, 16, $18 billion. But today we're making a big dent in the international market as far as agriculture and sustainable agriculture produce that we are producing for the communities, not only in India, but also for the other part of the world. Importantly, this is the only sector that I see which is producing trade surplus in the country. Farmers are facing problem because of the climate change induced changes that are happening. Also international trade lackluster in our whole quarantine systems, that we have seen influx of invasive species in last 10 to 15 years in the country, damaging prospects for production of corn or cotton or chili, whatever they produce that we feel that are important for the export markets. I believe in 10 years' time, we would see a huge transformations where country would move from 5,000 liter per kg rice production, water that we need, to maybe less than 500 liter water per kg of rice that we can produce. So there are technologies, some mutant lines which are tolerant to herbicides, popular herbicides are being now approved by the government of India. And we see that in future, you will see the rice that are producing, which is much more efficient and less use of water. And therefore, we can continue to be competitive and continue to be a country that can supply rice or other food crops that meet the SPS conditions. So possibility of collaboration beyond institution in India with international communities. We looked at European Union as one of the partner, being a G20 member country, who can actually share expertise on food safety, food processing, Codex compliant MRLs, on pesticides, because they take a lot of these unilateral decisions that harm exports of agriculture commodity from India. And also the whole possibility of traceability and certifications. We looked at Argentina as a country that can actually collaborate with India on drought-tolerant HB4 wheat. So this is a big innovations which provide the tolerance to drought in water stress conditions. Brazil, of course, we are working very closely on cotton and mosaic virus resistance beans. US has big expertise in drought tolerant again, and the fall armyworm resistance technologies in corn. Similarly for Canada and Indonesia and South Asian, with some of the South Asian countries.
Interestingly, we came across a very unique microbial community in the desert, which is the biological soil crust. So you can think of the biological soil crust as a functional skin of the soil, which is formed in the top few centimeters of soil by the coordinated action of several microbes, predominantly cyanobacteria, algae, microfungi, lichens, bacteria, and bryophytes. So the soil particles are aggregated together through the presence and the activity of these microbes. And just imagine cyanobacteria out in the desert in an arid zone. Who would have thought? That was our first reaction when we first heard about it. And then we learned that the crust has a microbial community that has adapted perfectly for survival under arid zone ecosystems. Now, how does it do that? We know that the desert is a very resource-constrained environment, and typically resource-constrained environments will push microbes to cooperate because individual survival is not possible. There's extremely high levels of radiation, so you need some kind of protection from UV, and there will be prolonged spells of aridity, which might mean that you need some kind of protection against desiccation, high temperatures, of course, and increased salinity in the desert, and variable pH across the soils. So the entire community must adapt to survive in each of these functions if it has to make a living in the desert. So this is an exciting community, of course. It's interesting, but you might ask me, why do I care? Why do I care about a soil microbe that lives in the desert? Turns out, these communities perform key ecosystem functions that are required for soil formation, soil development, which will ultimately lead to better growth of vascular plants in the desert. So if you think about it, the cyanobacteria in the crust can fix both nitrogen and carbon through, of course, photosynthesis and nitrogen fixation genes. They participate in biogeochemical cycling, in the process of which they form the soil itself. The cyanobacteria also secrete these polysaccharides, which help kind of stabilize the soil and prevent erosion, and also trap dust, which also acts as an additional source of nutrition. So in fact, biocrusts have been thought of the predominant carbon sinks in the desert next to vascular plants, in the sense that we're going to be losing these microbial communities of the desert, which are shaping up the desert ecosystem. So they are additionally also threatened by uninformed greening, agriculture, and other human activities such as mining, tourism, etc. So when we started getting into this problem, we wondered, how do these crusts form? It's actually a very interesting and elegant process that happens. The pioneer species that gets onto the soil is actually cyanobacteria. And these are not normal cyanobacteria, these are actually filamentous cyanobacteria. So when they hit the soil, they start photosynthesizing and making these polysaccharides, and they slide in and out of the soil. Why? Because there's too much UV on top, so they need to go burrow under the soil to protect themselves from the UV. And when there is water or a hydration event, they would move up through these channels of polysaccharides and photosynthesize and then rapidly go back inside. So this process of moving up and down all the time secreting polysaccharides actually makes an underground network of polysaccharide fibers, which is the initial step of biocrust formation, which kind of stabilizes the soil. So the species that's involved is actually Microcoelius vaginatus. This is, of course, information from what's available in American deserts. There's no data available for the Thar Desert itself. So what you're seeing on the right is actually the series of events that would happen. You would have bare soil in the beginning. Within a few months, slowly the soil would first get stabilized. Then over time, you would start to see pigmentation, which is actually the overgrowth of these cyanobacteria. So this is a small portion of the repository that we have established from biocrusts. We were able to isolate cyanobacteria which are filamentous and growing in clumps, as well as unicellular cyanobacteria. We were also able to see that biocrust microbes are producing antimicrobial substances, which might be of value to the industry. How do we access the diversity of the microbes? We turn to shotgun metagenomics, which is enabled by nanopore sequencing. So the idea is to extract DNA directly from the soil sample, biocrust sample, fragment the DNA, and then read it using a DNA sequencer. And then from the genomes, try and understand what the microbial community is and what functions these microbes are doing there in the environment. Going forward, if you look at the species of cyanobacteria that are present in the crust, what is evident is it looks like the amount of moisture that's present in the particular environment. It's consciously that we picked up crust samples from the wet, a semi-dry, and a dry environment. What you see here on this far corner is basically the Akal rocky soil crust, which has a distinct microbial community. And this is the one in Kanoi, which is, of course, the grainy soil of the desert. They are very well separated from their respective control soil samples. And we see a group of samples here, all of which were collected from wet Thar, Beriganga, and one wet area in Akal. We also see that within the cyanobacterial population, the Microcoelias that we were talking about earlier, which is the pioneer species, seems to be quite well distributed, but it largely seems to be restricted only to the wet areas of the Thar. So that means that our biocrusts that are forming in the Thar are not really similar to biocrusts that are seen in other deserts. We are also seeing that some species like Cytonema species and Allocoleopsis, which are enriched in crust samples, appear to be specific to this local ecoregion. I want to talk a little bit more about the Akal rocky biocrust because it's basically life on nothing, right? There's very little nutrients available. So we see that this particular crust, the cyanobacterial populations are largely Cytonema species and Crucoccidiopsis species, both of which are pigment producers, heavy pigment producers in the sense that it offers them protection from the UV, and they can actively photosynthesize as well as tolerate extremely high amounts of radiation as well as desiccation. In fact, this particular species, the Crucoccidiopsis, has been tested on the International Space Station as an astrobiology experiment to see how long it can survive in space, and has also been proposed to be used in the terraformation of Mars. So where to now? So we would like to further understand how biocrusts develop in these areas, and would also like to use remote sensing and other approaches to map the spread of biocrusts in the Thar Desert and try and guide policy on how biocrusts as a resource have to be conserved, as well as try and restore the biocrusts which are damaged by anthropological activities using the biocrust biorepository that we have constructed in the laboratory.
topic is how marker-resisted breeding is useful for the development of resistant varieties. So I'll start with this slide. This slide depicts the present agricultural scenario in India. The graph indicates the food production scenario right from 1950 anyway, since from independence to the present day, from 50 million ton food production to 315 million tons. It's a huge achievement. And India has become from food deficient to we are now food sufficient, as well as we are already exporting different foods, food products and commodities to the other countries. It is a more than 6 times jump in the food production. And not only in food production, we are also having a significant achievement, like 13 times increase in the horticulture produce. That comes under fruits, vegetables and all. Oilseeds, that is again 6 times jumps. Pulses, 3 times jump. Milk production, we are the largest producer in the world. Milk as well as meat, livestock, eggs, everywhere. India has achieved a significant achievement. Although we are still lacking some edible oil and pulses, still we are importing from outside. But this is a tremendous achievement in the field of agriculture. So as you know, the great achievement comes with the biggest challenges. So there are several challenges. In the previous lecture, Dr. Bhagirath also had discussed, but climate change is one of the serious challenges that is present in our doorstep. We need to really think of it, because all the predictions says that the climate change will directly impact the agriculture production and our food security. So it will change the microclimate as well as the macroclimatic factors that will directly or indirectly lead to the agriculture production, quality as well as quantity. And most importantly, it will also change the pest and pathogen dynamics. And present day, in the whole world, if you see the record of FAO, more than 3.5 million tons of pesticide, I'm talking only about the pesticide, are being used in the agricultural field. In India also, we are not less behind. More than 61,000 metric ton of pesticides we are using annually. And these pesticide residues, they are contaminating the groundwater, they're contaminating the soil, environment, our foods, products, as well as our health. So developing resistance varieties can be one of the solution for the farmers to reduce the pesticide use, reduce the production costs, also increasing the profitability of the farmers and bringing the sustainability in the agriculture. How molecular markers or marker-assisted breeding is useful. There are several use of molecular markers, but to discuss some of these, like for early detection of the plants, even before the onset of the disease, molecular marker can help us to select at the seedling stage whether the plant will be resistant or it will be susceptible. Dealing with the quantitative traits is one of the biggest challenge in the breeding, because in case of quantitative traits, we are not able to differentiate the resistance as well as the susceptible alleles by looking at the phenotype, because there will be minor difference in the phenotype. So molecular markers helps us to select the plants with the positive allele or the resistance allele. Again, when we need to pyramid the multiple genes of the same disease or different diseases, in that case, molecular marker is very much useful. And speedy record of the genome. If you want to improve a particular variety, and by keeping all the quality of the variety as such, we need to recover the background genome. In that case, molecular marker is very useful. This one slide that I'll show how marker is working. The upper two picture, that is picture A and picture B, there is infected plant with leaf rust. That is wheat plant. The variety is BRW934. And the below picture, that is Chiria 3, that is resistant to the leaf rust disease. But it is carrying one of the resistant genes, that is LR34. So if you look at the picture D, where leaves are shown, Chiria 3, the leaves of Chiria 3 is clean. There is no disease, but its tip is necrotic. It's dried up. And the susceptible variety, the whole leaf is already infected. Now, genetic study shows that the gene for tip necrosis is present in close proximity with the LR34 gene. So now in this case, tip necrosis can be used as a marker for LR34 gene. Similarly, DNA-based marker, this is a agarogel product of one of the marker that is CSLB34. CSLB34 says that the shorter band are in close proximity with the resistant allele of LR34, and the larger band is associated with the susceptible allele. And these are the band of the hybrid products. So by using this marker, we have transferred the LR34 gene to the susceptible variety BRW934 from Chiria 32, BRW934, and you can see the two plants, that is progeny of BRW934 that is carrying LR34 gene, showing resistance reaction, and without LR34 gene, it is susceptible reaction. The presence of LR34 is reducing also the disease severity significantly. This is another case, and wheat blast disease. There is very recent disease identified in first time in South America, in Bolivia. Is one of the most dangerous diseases in the wheat because it can infect the whole plant. Once it affects the ear, where the grains are formed, it's called ear. Once it affects the ear does not form any seed grain. It will be totally chuffy. And under favorable climatic condition, the disease causes 100% yield loss. So really this disease is very dangerous. And from Bolivia, it has transmitted to the adjoining countries like Brazil, like Paraguay, Uruguay, even Argentina, and it causes a huge loss in the wheat production. Now, what is our concern? In the year 2016, the disease appeared in Bangladesh. Now, that causes a serious concern for us because the whole central India and parts of the Pakistan, there is favorable condition for the disease. And government of India has taken several steps like declaring wheat holiday in the adjoining districts of Bangladesh, where in the West Bengal states, there will be no cultivation of wheat for 5 kilometers border area, strict quarantine measures, and also it shows that the color indicates the migration of the disease from different districts year by year, from 2016 to 2021. So what we did a study of Indian wheat germplasm by screening them in Bolivia, and we found that some of the genotypes are resistant, and we tried to map the resistant locus that is present on the terminal end of the 2A chromosome, and it is the translocated segment, it is known as 2NS. And this variety we have developed, that is BRW3806, it is highly resistant to the wheat blast disease. And it has observed that the presence of 2NS can significantly reduce again the blast index. But unfortunately, most of the cases, this is the only resistance source found for the 2NS, either from Indian germplasm, from Bangladesh germplasm, even from Chinese germplasm, Mexico, South America, even US germplasm. This is the only locus that is showing the highly resistance to the 2NS. So there are a few cases where marker selection cannot directly useful for transferring the locus or gene. This is one of the typical case for spot blotch disease. If you see the chromosomes, this indicating the different locus or the markers that is showing the resistance to the spot blotch, and the locus as well the QTL, they are spread throughout the genome, and none of the QTLs are major QTLs. So in case of minor QTLs, it's very difficult to transfer to the mass, and in that case, genomic selection can be very useful. Where phenotyping is also very difficult, where markers are present throughout the genome. So genomic selection can play a very significant role in that case for developing of varieties. So we did a model study for spot blotch, and we observed that genomic selection, the genomic prediction accuracy 0.53 across the panel where there is no relatives, and within panel where there is some genetic relationship between training as well as the breeding population, the genomic prediction accuracy of 0.4. But this is not so high for mass till date. But one important thing but the red dot indicates that in most of the cases, more than 90% of the lines that are susceptible to the disease, we can reject it before testing to the field. So it will be very significant achievement by rejecting the susceptible lines from testing, evaluating to the fields, and it will reduce the time as well as the cost. So these are the few other examples. In case rice, peanut, wheat, and millet and maize, though there are several others, where marker-restricted breeding was used for developing resistant varieties. And then I would like to say, most of the cases marker-restricted breeding was followed in major crops, where more number of molecular markers are available, where fundings are available, where research groups are available. But we need to really look into the crops which are locally available but very high value crops are very important, like fruits, like vegetables, like spices. We need to think that how these crops can be come into the market selection. And at the end, I would like to thank to the organizer for giving me the opportunity to share my thoughts here. Thank you very much.
So I am going to talk about abiotic stress management. So this is a part of the One Health. So my friend, Dr. Chandan Roy, talked about the biotic stress. So I am Dr. Ayun Sadhu Khan. I work in plant population genetics. I use Arabidopsis model and I look into the plant cell at the molecular level, what's going on. And also we study about the molecular markers in Arabidopsis and how that, and genome-wide association study, how that can lead to discovery of genes for abiotic stress tolerance. So this already have been talked about. We are just crossed China and the most populous country in the world. And there is this problem of food security. So apart from biotic stress, the abiotic stress or the environmental factors like drought is one of the major constraints of agricultural productivity. And we are rapidly progressing towards land degradation and desertification. So among other measures, the improvement of plant productivity under abiotic stress, that is more crop per drop, is the global agenda. So we have to understand the molecular mechanisms of plant abiotic stress tolerance to develop varieties which are more robust. So this drought, UV, salt, heat, cold, this kind of abiotic stresses are complex quantitative traits involving many genes and complex gene networks. So the signal is usually perceived by certain ion channels and transmitted via secondary signaling molecules to transcription factors. So there are actually stress-specific transcription factors. For example, the well-studied dehydration responsive element binding protein, DREBs, so which ultimately bind to the promoters of certain downstream genes, which lead to ion homeostasis, scavenging of ROS, protection of the cell structure, etc., under abiotic stress. Now there is a catch. The single drought stresses are very rare in the field. They actually come after intermittent water recovery intervals. So while a severe drought may be detrimental for the plant, mild drought can be tolerated. And in the next drought episode, the plant will present a more robust response. So there are two types. One is the transcriptional or short-term memory. Another is the transgenerational memory, which can even be transmitted to the next generation. So we are beginning to look at the molecular mechanisms of memory. So apart from being sessile, plants also have this capacity of memorizing the stress. So we can see here in the figure, right-hand side. So there is the watered interval followed by a first drought, and then the plant receives some rainfall, for example. In the second drought, you can see some genes are progressively upregulated, while some genes are downregulated. So here you can see this kind of transcriptional memory in case of this ABA and J biosynthesis genes. So these are the drought-specific hormones. So their biosynthesis involves this transcriptional memory. So we can see in the green part of the figure the stomata. So when there is a fast drought, the plant will close the stomata rapidly, but it will memorize that response. And when in the next recovery interval, the plant will not open the stomata fully. It will remain partially open. So this is a strategy so that it can very quickly close it in the next drought. So these are the one of the newest findings. We are just beginning to appreciate this memory behavior in plants. And these are very important in development of stress-resistant varieties, which can actually memorize the stresses. So why are these desert plants important? So these plants, desert plants are mostly medicinal plants. We know they have a lot of secondary metabolites. These are actually designed for them to survive these stresses. While we uptake them as the medicines, they help us fighting our loss and curing our diseases. So these desert plants will contain tolerance genes responsible for these abiotic stresses and can be the sources of for bioprospecting. So they are the sources of novel mechanisms for integration into the cultivated plants by means of biotechnology. So we can see here the two types of, one is invasive Prosopis juliflora and another native khejri or Prosopis cineraria. So we can see this polyploidy. The polyploidy plays an important role in the evolution of legumes. And the legumes are, of course, the most tolerant plants. So we can see the polyploidy or 2n equal to 56 in the juliflora, so in the invasive plant. So you can see in the map that this invasive plant is actually native in the South America. So there is a question why this invasive species outgrow and this Prosopis. So we can, one study has been done. Here it has been found that this soil microbiota, so that is called rhizosphere, the soil around the roots. So they, in case of juliflora, the invasive one, the beneficial actinobacteria in the rhizosphere of those plants actually produce some specific antibiotics. They kill the plant pathogens. So more than what the beneficial microbes in the khejri or native cineraria cannot do. And we work on this model Arabidopsis. So Arabidopsis is a very small plant. It's like the mouse of the plant biology. So it has a very short life cycle. It is self-pollinated, and there are a lot of data. So we have this Arabidopsis from around the world, different ecotypes or strains, as well as several knockout lines collection. And we have around 1 crore, more than that, SNP information, the genotyped data. So this Arabidopsis actually reflects the higher plants, so we can do a lot of fundamental research in Arabidopsis, so which can be translated later into the field crops for higher yield and stress tolerance. So we use GWAS or genome-wide association study as a forward genetic tool for gene discovery. So we are also bioprospecting several plant growth promoting bacteria from the Thar. So Thar desert is a huge resource, so which can have direct applications in arid agriculture. So we have actually from the rhizospheres of several desert shrubs. One is Tephrosia sarfunkha, plant described in Ayurveda. So we have isolated several strains. So you can see in the figure that a plant growth is drastically improved by this plant growth promoting bacteria. We have actually gone to the extent of sequencing the entire genome of this bacteria. And we can see even in the pig, the drought stress, there is a drastic improvement in the root and shoot growth. So this genome contains several genes which have these plant growth promoting traits. For example, auxin production, it can produce auxins, which lead to the longer root, as well as several genes for nutrient solubilizations from these nutrient poor soils. So another approach, so this is the culturable approach, what Professor Shankar talked, but there is another approach like this functional metagenomics, where we clone directly the genes and genetic cassettes from the soil samples from the rhizosphere and use for development of stress-tolerant plants. So we are also bioprospecting these drought resistance genes from the khejri, the Prosopis cineraria. We have grown these seedlings in the lab and mimic the drought stress, and we have done the whole genome like transcriptome sequencing, and we have identified the genes for drought resistance. So
field, so I'll discuss about the Ayurvedic perspective of the climate change also. our acharyas have told about this climate change, and they have foreseen about this climate change thousands of years ago, and they have told it under the heading of Janpadadvans. They have said that when the four factors, that is Jal, Vayu, Desh and Kal, when these all get vitiated, then definitely the stage will come of the Janpadadvans. The Janpadadvans is all about the epidemic diseases, and it is correlated by the climate change. And the fourth factor, that is Jal, Vayu, Desh and Kal, the Kal is, in this scenario, is the climate change. The Kal Viparya is the Janpadodvans. It is leading to the number of epidemic diseases. Acharyas have also said that very soon the earth will cease to manifest proper taste, potency, etc. So Lord Acharya had advised Agnivesh to collect herbs before the time of destruction and before the earth loses fertility, leading to impairment of taste, potency, vipak, and special effects. So Acharya had told to preserve the seeds before the complete. The Ashtavarga consists of Jivak, Rishvak, Meda, Mahameda, Kakoli, Shir Kakoli, Riddhi, and Riddhi. Nowadays, these drugs are unavailable because all these drugs are grown there at the high altitudes, but they are also they are become extinct because of these climate changes. And so now in the medicine we are using substitute of it, which are Vidari Kand, Shatavari, Ashwagandha, and Varahi Kand. And though we are using substitute, but the original effect of those drugs are not obtained till now. Ayurvedic methods to tackle climate change. Conservation of the biodiversity. All these are being told by our acharyas many thousands of years back. The conservation of biodiversity, sustainable harvesting, over exploitations. So medicine plants from the climate change, which has been told by our acharyas, and in modern time it also has been said that the seed banking is there, which is told by our acharyas also. The biotechnology, the genetic engineering is there, and research on phytochemical changes happen to plants should also be promoted. The prevention of biodiversity loss, I have already discussed about it, and breeding approaches should also be promoted.
Technological solution at computer science level, we always think about IoT. So I'm going to talk about IoT, Internet of Things, where everything is connected to internet so that we can monitor them or observe them or maybe respond to the changes or plan for the changes. So like in IoT approach, we'll make our complex system simpler. We are trying to do that in our lab. So we'll talk about that part. Our problem when we need IoT, our human senses are limited. I'll give you a very naive example. We can see less than 2% of the whole radiation, and we can hear less than 2% of the whole waves. So it makes that 98% of the world is not visible to us. But here our sensors came in action. So we can use sensors which we can't see or which we can't perceive. Using these sensors, we can measure lots of parameters like we are doing in our clinical labs. Our doctors are using those parameters to treat us. So that is about only human health. So we have body area network where we talk about these various sensors and try to capture those parameters, maybe continuously with real-time monitoring and maybe remote monitoring using telemedicine solutions. So there we need IoT for human health. Of course, there's a later part of the network where we want to transfer these values in real time without compromising security or privacy breach. So those all complexities are also there in the system. Similarly, we have sensors for plants. So like precision agriculture, or we want to monitor, say, what Professor Shankar talked about the bacteria growing on soil surface. If we want to monitor remotely, not every one of us can go to Akal or go to Jaisalmer every day and collect the samples. So we need remote solutions to have continuous monitoring and have early intervention when it's required. So there are various ways, various kind of sensors like UAV sensors and humidity sensor, temperature sensors, which can be connected with the system and can be used for continuous monitoring. Then we have IoT solution sensors for animals as well. Generally, we do in counting or tracking the path of migratory birds from one location to another location. In fact, we every year we deploy large number of sensors in high altitude areas in Leh, Ladakh, and Kashmir areas, and so that we can closely monitor the environmental condition and accordingly respond. So majorly Army uses those sensors as of now, but we now at this point of time we can start thinking not only Army, we should use those sensors data also for One Health perspective. So I explained why IoT is required and what IoT can enable us. Here is small thing which, not small thing, it's a complex small thing which we do in our lab. We build solutions which are IoT enabled. There are various types of sensor with heterogeneous data with their different sampling rate, different quality quality parameters, and we want to aggregate them all together. You can imagine it is not easy to have 5 people in a meeting like we see in panel discussion. 5 people sitting there and all of have different opinion. In the same way, the sensors which we are producing from our assembly line, they're also not exactly same. So when we want to aggregate those sensors value which are heterogeneous, it makes our task much more challenging. So data analytics and data processing becomes much more complex in that case. So when we talk about this complexity and at the large scale, you can see the web scale. Web scale system where all systems are interconnected, and still we don't have many things connected to internet at this moment. But now we are talking about the Internet of Things, which will outclass the web scale system. It will be larger than our current existing internet. So in those scenarios, how to tackle compute, how to tackle storage, how to tackle the communication part, which will have more complexity in optimizing the resources. So we work out this evolution part of connectivity, storage, and computation part, and we try to see how they are converging at this point, and in future what kind of technologies we can foresee for enabling or implementing IoT-based solutions. So once you can imagine we have IoT system up to certain scale. Now the next problem comes about the data processing. Simply having a data which is being generated by say thousands of sensors every second if it's generating one data point, and all those sensors are geographically apart from each other. Having a connectivity among them and ensuring the robust, reliable connectivity, it's not a straightforward task. I'll give you one example. The Wi-Fi which we are using, it has 37% accuracy on an average. Means the data you are transferring, so you are transferring 1 GB data. Actually, you are transferring 3 GB data. Out of that, 1 GB is correct. 2 GB was erroneous, so we discarded it. And you can perceive it while you are using mobile data and downloading some say 200 MB file, and you see your internet usage goes to 600 MB because your 2/3 of the data was not correct, so your system has discarded it. Now we are talking about the large scale system, and if we are working with 1/3 efficiency, that's not going to work. It will not be cost optimal. So there are various ways in which we can maintain this large amount of data with heterogeneous data which I talked about, like it can be text, it can be images, it can be videos, it can be audio files as well. So there's a multimedia data which we would like to process it. So different type of data, different nature of data will suggest us to use different approaches like graph processing or textual processing or incremental learning. It depends on what is the context and what kind of data we are collecting. Then we have data analytical category. You can imagine so far we have transferred our movie from USB to our laptop. That was data processing. Now we actually want to watch movie. So when we play movie, high definition movie, there lots of compute resources are used to watch the movie. In the same way, after data processing, when we jump to data analytics, we want to answer some unknown questions. Then data analytical frameworks come into picture. And here we start with traditional statistical and mathematical approaches to more heuristic and deep learning based approaches. Now we are shallow learning, hollow learning as well. So large, here again we have heterogeneous approaches for different contexts. Now you can see there's lots of heterogeneity in IoT based system if we want to analyze the data. So complete pipeline from start to end is quite large and complex. So we need some robust and highly scalable solutions, which we don't have as of now. And in our lab, we are continuously working on such systems and making. So we are working in a resource constraints environment where we have small machine and we can scale our system to thousands of millions of users with a small laptop kind of system, the commodity hardware. These kind of solutions will enable us in future to scale our IoT for One Health. I'll give you one problem which we generally face while building such system is fault tolerance. Suppose there is any failure in any component or any entity or any part of the system, and we want to handle that fault. We don't want our system to fail because of that one or two or partial fault in the system.